Alanine is an I plus or minus amino acid with the chemical formula CH3CH, NH2, COOH. The L-isomer is one of the 20 amino acids encoded by the genetic code. Its codons are GCU, GCC, GCA, and GCG. It is classified as a non-polar amino acid. L-Alanine is second only to leucine in rate of occurrence, accounting for 7.8% of the primary structure in a sample of 1,150 proteins. D-Alanine occurs in bacterial cell walls and in some peptide antibiotics. Structure: the I plus or minus carbon atom of alanine is bound with a methyl group, making it one of the simplest I plus or minus amino acids with respect to molecular structure and also resulting in alanines being classified as an aliphatic amino acid. The methyl group of alanine is non-reactive and is thus almost never directly involved in protein function. Sources: dietary sources. Alanine is a non-essential amino acid, meaning it can be manufactured by the human body, and does not need to be obtained directly through the diet. Alanine is found in a wide variety of foods, but is particularly concentrated in meats. Good sources of alanine include animal sources, meat, seafood, casein, dairy products, eggs, fish, gelatin, lactalbumin. Vegetarian sources, beans, nuts, seeds, soy, whey, brewer's yeast, brown rice, bran, corn, legumes, whole grains. Biosynthesis. Alanine can be manufactured in the body from pyruvate and branched-chain amino acids such as valine, leucine, and isoleucine. Alanine is most commonly produced by reductive amination of pyruvate. Because transamination reactions are readily reversible and pyruvate pervasive, alanine can be easily formed and thus has close links to metabolic pathways such as glycolysis, gluconeogenesis, and the citric acid cycle. It also arises together with lactate and generates glucose from protein via the alanine cycle. Chemical synthesis. Racemic alanine can be prepared by the condensation of acetaldehyde with ammonium chloride in the presence of sodium cyanide by the Strucker reaction, or by the aminolysis of 2-bromopropanoic acid. Physiological function. Glucose-euroalanine cycle. Alanine plays a key role in glucose-euroalanine cycle between tissues and liver. In muscle and other tissues that degrade amino acids for fuel, amino groups are collected in the form of glutamate by transamination. Glutamate can then transfer its amino group through the action of alanine aminotransferase to pyruvate, a product of muscle glycolysis, forming alanine and I plus or minus ketoglutarate. The alanine formed is passed into the blood and transported to the liver. A reverse of the alanine aminotransferase reaction takes place in liver. Pyruvate regenerated forms glucose through gluconeogenesis, which returns to muscle through the circulation system. Glutamate in the liver enters mitochondria and degrades into ammonium ion through the action of glutamate dehydrogenase, which in turn participate in the urea cycle to form urea. The glucose-euroalanine cycle enables pyruvate and glutamate to be removed from the muscle and find their way to the liver. Glucose is regenerated from pyruvate and then returned to muscle. The energetic burden of gluconeogenesis is thus imposed on the liver instead of the muscle. All available ATP in muscle is devoted to muscle contraction. Link to hypertension. An international study led by Imperial College London found a correlation between high levels of alanine and higher blood pressure, energy intake, cholesterol levels, and body mass index. Link to diabetes. Alterations in the alanine cycle that increase the levels of serum alanine aminotransferase is linked to the development of type 2 diabetes. With an elevated level of ALT, the risk of developing type 2 diabetes increases. Chemical properties. Free radical stability. The deamination of an alanine molecule produces a stable alkyl free radical, CH3Ca-HCOOA-. Deamination can be induced in solid or aqueous alanine by radiation. This property of alanine is used in dosimetric measurements in radiotherapy. When normal alanine is irradiated, the radiation causes certain alanine molecules to become free radicals, and, as these radicals are stable, The free radical content can later be measured by electron paramagnetic resonance in order to find out how much radiation the alanine was exposed to. Radiotherapy treatment plans can be delivered in test mode to alanine pellets, which can then be measured to check that the intended pattern of radiation dose is correctly delivered by the treatment system. See also, carbamic acid, glycine, references. External links, alanine mass spectrum.